Welcome back, everyone, to now the 13th installment of Emerging Topics in Biomolecular Magnetic Resonance. And as always, this is some information that is available for older lectures that have been recorded and can be found on the web at the following YouTube channel. And of course, we remind everyone that we have a Gordon Research Conference policy of no private recordings, which is anyway not necessary since we'll be recording the lectures today. For those who are new, at the end we'll have a Q&A, so please type your questions in the Q&A if you'd like, with your name attached or anonymously, so that people can vote on your questions. Or alternatively, we can also, if you raise your hand, we can also select you to speak, so that it's a little bit more interactive. So with that, it's my pleasure today to introduce Sang-Ho Park, who is currently a project group leader with Stan Opella and University of San Diego. And I'll just read a little bit his background, coming from a PhD in Seoul National University in South Korea, a master's there as well, and then going on to San Diego as postdoctoral work with Dan Opella, where he is now. And the interests of Sang-Ho Park are mainly in membrane proteins, with a lot of impressive work with different kinds of alignment, from rotationally aligned samples to more traditional aligned samples, and more recently some interesting work with macro disks. And so today we'll hear, I guess, about some very emerging topics in membrane proteins, proteins coming from, of course, the coronavirus, in particular the E and S proteins today. So with that, the floor is yours.
Thank you. Thanks for the introduction. Okay, so I also like to thank the organizers for inviting me to this wonderful webinar. Since COVID-19 pandemic hit, so our group started thinking about how to fight against it using NMR. And since we have studied the vital membrane protein VPU from HIV-1, so we got immediately interested in structural membrane proteins in SARS-CoV-2. So we started cloning as soon as the lab reopened, which is early this summer, and then have been collecting NMR data since then. So the result I'm presenting today were obtained with the support of Opella Group and also our collaborator, John Guatelli's group at UCSD Medical School. So SARS-CoV-2 RNA genome consists of about 30,000 nucleotides and encoding 29 proteins. So the 4 structural proteins, along with the vital membrane, form a characteristic, this characteristic SARS coronavirus structure with about 120 nanometer diameter. And they protect their genetic material inside. And other 16 non-structural proteins and the 9 accessory proteins are found in the infected host cells. And these 4 structural proteins called MENS, membrane protein, envelope protein, and nucleocapsid, and spike protein. Can you see my cursor? Okay. The 3 of the structural proteins are membrane proteins, except for nucleocapsid. The M protein has 3 transmembrane helices, and E and S protein have the single transmembrane. And all the membrane proteins have been shown or proposed to exist as oligomers. A dimer or dimer of dimer for M protein, a pentamer for E protein, and the trimer for S protein. And S protein consists of 2 main subunits or a domain, receptor binding domain and the membrane binding domain. And the structural proteins are involved in extensive protein-protein interactions. Not only are there vital host protein-protein interactions, but also the many intravital protein-protein interactions. For example, the C-terminal region of the E protein interacts with the C-terminal region of the N protein and S protein, and potentially with the nucleocapsid protein too. So right now we are working on all three membrane proteins, but today I'm going to focus on mainly E protein and show some initial result on the transmembrane domain of the S protein. So E protein is the smallest of the structural protein, but it plays an essential role in assembly, budding, envelope formation, and pathogenesis in the coronavirus life cycle. The E protein of SARS-CoV-2 is 75 residue-long polypeptide with a single transmembrane helix and then 3 cysteines right after. And the sequence is actually almost identical to original SARS coronavirus, which appeared in 2002, except for the residues shown in green here. And the transmembrane domain of E protein has been reported to oligomerize and function as a vital protein, which is a virus ion channel. And the C-terminal domain of E protein is mainly responsible for protein-protein interactions, both intravital and host-vital interactions. And especially the 4 C-terminal residues, DLLV, it was identified as a PDZ binding motif. So it is important for host-vital interactions. And recently, it was found that this motif is highly conserved among the all beta coronaviruses, which SARS-CoV-2 belongs to. So it is important to keep the C-terminal domain intact for structure and functional studies. So recently, our collaborator, the John Guatelli's group at UCSD Medical School, has shown that the SARS-CoV-2 E protein significantly increases the virus-like particle production. The virus-like particles, or VLPs, self-assembles with only structural proteins without its vital genetic material. So they are not infectious, and they are used for studies of virus structure, virus-host interactions, and also vaccine development. So here in cell lysate, which corresponded to the infected cell lysate, the presence of E protein does not affect the expression of the structural protein N here. However, in the supernatant, the same protein in the presence of E, the same N protein expression was significantly increased. Due to the increase in VLP production. So again, indicating that E protein plays an important role in the SARS-CoV-2 life cycle. Many coronavirus E proteins have been shown to form the cation selective ion channels in lipid bilayers. And on the left is the a plaque assay showing the chemical the channel blocker hexamethylene amiloride, HMA, but not amiloride, inhibit the replication of the mouse coronavirus and also human coronavirus. And the right is the Pantama model, and the HMA binding studies of SARS-CoV E protein has been reported by NMR with the E protein construct A265, which has 26 extra residues at its N-terminus, and all cysteine were replaced with alanines. So, however, it is essential to have the intact full-length E protein for studying protein-protein interactions. because as I mentioned earlier, the C-terminal domain of E protein is mainly responsible for intravital and the host vital protein-protein interactions. So, based on our extensive experience with the sample preparation of membrane proteins for NMR studies, we have designed 3 E protein constructs. The full-length E protein, we call it EF, with all cysteines intact, and the transmembrane domain of E protein, ET, and also the cytoplasmic domain of E protein, EC. Again, the 3 cysteines are intact. And we place 10 histidine tag followed by thrombin cleavage site between the fusion partner, keto steroid isomerase, KSI, and our target E protein construct. So the KSI boosts the protein production in E. coli, and this long flexible loop facilitate the protein purification by using nickel affinity chromatography as well as thrombin cleavage. So this single expression and purification system was successfully applied to all these E protein construct, and the protein sequence is intact with only 2 extra residues, that is N-terminus. And here's the SDS page showing the expression and purification of full-length E protein. The E protein fused to KSI was expressed very well in E. coli as inclusion bodies as expected, and this inclusion body was solubilized in mild detergent, hexadecylphosphocholine, or HPC, or phosphocholine 16. Then applied to the nickel affinity chromatography, and we purified the fusion protein here, and this fusion protein cleaved with a thrombin, and this thrombin cleaved product reapplied to the same nickel column, and we collected pure E protein, full-length E protein in the flow-through. Then we directly concentrate this pure E protein and examine the NMR spectrum. The same protocol was applied to the truncated ET and EC construct. The overall yield was pretty good. It was about 12 milligrams per liter culture for N15 uniformly labeled protein in M9 media, and 6 and 10 milligrams for ET and EC, which is sufficient for NMR experiment. And the whole purification procedure was very efficient and takes 2 days, including overnight incubation for thrombin cleavage. The mild detergent, the hexadecylphosphocholine, was used throughout the procedure and also for NMR sample preparation. So the protein was never exposed to harsh conditions such as strong acid for chemical cleavage reaction or organic solvent for HPLC purification. And here's the HSQC spectra of 3 E protein construct in HPC micelles. In the middle, the full-length EF spectrum, and on the left is the transmembrane ET in red. On the right is the C-terminal domain E protein EC in blue. So you can see the chemical shock dispersion of amide protons are typical for helical structure, which is about 1 ppm, but overall they are very well resolved. And the signals of ET spectrum are observed mainly a downfield region relative to the APPM and overlaps very well with the full-length protein. And then the C-terminal domain also overlaps very well with the full-length. So this result indicate that conformation of truncated ET and EC are conserved. And all these spectra set the stage for characterizing the SARS-CoV-2 protein by NMR. And these are the spectra of EF, transmembrane, and then full length. exchanged with the D2O, and 15 signals survived in full-length residues from 17 to 31, which corresponded to the core of the transmembrane helix, whereas 13 signals were observed in the transmembrane domain ET, residues from 17 to 29. So the threonine 30 and leucine 31 were exposed to exchange to D2O in the absence of the C-terminal domain. Okay, so then we did the drug binding experiments on full-length E protein. This is the 1DN15 edited proton chemical shift spectra of E protein in the absence and in the presence of channel blockers, the amiloride, and then 3 amiloride derivatives, dimethyl amiloride and ethyl isopropyl and hexamethylene, HMA amiloride, with a drug to protein molar ratio of 10:1. And we noticed that a chemical shift perturbation of E protein in the presence of drugs are mainly in the downfield region relative to APPN, which is primarily the region of the transmembrane domain. And we also noticed that no perturbation was observed in the presence of amiloride, but the most significant perturbation was observed by HMA. And the perturbation by DMA and EIPA are in between them. So in the 2D spectra, again, in the full length, we can clearly see the pattern of the chemical shift perturbation by drugs. So no changes were observed by amiloride, and the medium change by DMA and EIPA, and the largest change by HMA. And the perturbation pattern seems to correlate with the hydrophobic functional group, shown here, a group of the amiloride. So the bigger the functional group is, the perturbation, the larger the chemical shift the perturbations are. So there are over thousands of amiloride derivatives developed so far. So it will be interesting to look into other amiloride derivatives by NMR in the future. And then we compared the full-length drug interaction pattern with the truncated transmembrane and then cytoplasmic interaction. So you can see the transmembrane domain is mainly responsible, mainly affected by the drug, and its binding site, as shown in, as highlighted here, is almost identical to those of the full-length protein. And no significant perturbation was observed with the C-terminal domain, EC. So this result confirmed that the channel blocker HMA interacts with the transmembrane domain. But when we overlay the signals of ET, the transmembrane and full length in D2O, shown in blue, you can see no changes were observed in this blue, the transmembrane core region. Okay, so now we are in the process of assigning the spectra. So we expect a better picture of the drug interactions with the SARS-CoV-2 E protein, as well as the structure of the unmodified full-length E protein in a very near future. Okay, now last 3 or 4 minutes or so, I'd like to show some initial data of transmembrane domain of SARS-CoV-2 spike protein. Here's the cryo-EM structure of SARS-CoV-2 S protein, consisting of 2 main domains, receptor binding domain and the membrane binding domain. However, the structure around the transmembrane are missing because the transmembrane region is cleared, deleted for a sample preparation. But this unknown region of S protein residues 1128 through all the way to the C terminus contains the heptad repeat 2 sequence, HL2 sequence, which is very important for a post-viral membrane fusion process. And actually, the HL2 peptide itself, polypeptide itself, underlined here, has a very strong inhibitory activity of post-viral fusion. So we decided to study this important part of the S protein by expressing the transmembrane domain, including this HL2 polypeptide and the transmembrane helix. And here's the spectrum of transmembrane domain of SARS-CoV-2 S protein in the same expression, purification, and animal conditions used for E protein, the HPC mice cells, and pH 6.5, things like that. The spectrum is well resolved with the 3 tryptophan side chains shown here, 1, 2, 3, and then the transmembrane core in D2O, shown in red, includes 2 glycine residues in the transmembrane region. So we are planning to do the protein-protein interactions between the transmembrane domain of S and the E protein and its inhibitory activity SA2. And eventually we will look into E and S in membrane lipid bilayers by a solid state NMR. So we will reconstitute the E and S protein into detergent-free lipid bilayer disk system, and especially the peptide macro disk system we developed in 2011, and therefore we have 10 years of experience with this system. And this system is well suited for oriented sample solid state NMR studies, and they are fully compatible with the inhibition assay in the cells. And recently we also established the polymer-based styrene maleic acid macrodisk system for membrane protein studies by NMR. And these are the typical solid-state NMR spectra of membrane proteins embedded in polymer-based, SMA-based oriented macrodiscs. And all the samples are well-oriented with their lipid bilayer normal perpendicular to the applied magnetic field, as you can see in phosphorus NMR. And the protein structures are directly mapped on the PySema spectrum with their orientation-dependent frequencies. So we will apply this latest technology to structural membrane proteins of SARS-CoV-2 to determine their structures, orientations in membranes, and their protein-protein interactions in the membrane lipid bilayers. So with that, I'd like to thank our UCSD Research Group, Opala Group, and John Guatelli's group at UCSD Medical School. And thank you very much for your attention.
Thanks so much for sharing your latest results with us. I see already a number of questions in the Q&A. And so I'll start there. And for those of you looking for it, it's on the bottom right of your screen. Please go and vote, even if you don't have a question. So I'll start at the top. So Gloria asks, for purification of E, did you use reducing agent?
For purification, yes, for the binding, to improve the binding efficiency, we added 1 millimolar TSAP during the nickel column purification, which does not affect the nickel column, the binding, but improves the target protein binding to the nickel column. Yes.
Okay, and Neeraj then asks, what was the concentration of channel blocker drug? I think you said 10 to 1, but maybe in millimolar?
It is 10 to 1, so the protein concentration was about 0.3 millimolar to 0.5 millimolar. So it's the drug concentration is 2 to 5 millimolar.
Okay, and then Thomas Schmidt asks, the channel blockers had an effect on the trip side chain chemical shift, 9 ppm. Does that indicate that the drugs might interact with membrane surface, of which tryptophan might act as an anchor moiety?
E protein sequence does not have tryptophans.
So those tryptophans were from the cytoplasmic.
No. There's no tryptophan in the E protein. So that's
okay, it was just a shifted peak.
Yeah, that's a backbone signal. I thought, okay, let me go to the 1D. Yeah, this is not a tryptophan signal. This is the backbone signal. Okay.
Okay, yes. And then Yan Li asks, how about the oligomer state of the E protein?
Yes, the specific interaction of the drug to our E protein construct suggested that our protein exists as oligomer because it's a channel blocker. But to confirm that, we are working on using the size exclusion chromatography, native gel, and also by NMR. So
Nosy measurements, maybe.
Yeah.
And I also see another comment or question related to an earlier one. What was the concentration of blocker drug? Could the effect seen be due to perturbation of the micellar environment?
Okay, but here the C-terminal domain also has the detergents. These are all same condition, pretty much the same concentration of the protein, same concentration of the micelle, the detergent, and then also the drug. So you don't see any change in EC. So here I think the change is mainly due to the drug protein interaction, not by the drug micelle kind of non-specific interaction.
Related to that, do you have an indication of on or off rates yet from
that we have to, no, we don't know yet. We have to study that.
Yeah, many things remain to be done. Ashutosh Kumar asks, the number of peaks seem to be quite less compared to the number of residues. Which sections do you not see, or is it overlap?
No, it is actually it's the right number of signal that I can. Okay. We have
so
a data.
Right number, it's just the impression from the.
So we have we have a spectrum here. Let me show you. Here, this is the selective labeled spectrum. So there are 14 valine in the full-length sequence, and you can clearly see the 14 signals, and then 14 leucines, and you can see leucines, but leucine and valine overlaps here and there. So in the full length, it looks less signals, but there are right number of signals.
Okay, so everything looks like a symmetric pentamer, I suppose, then, so far. Then Gennaro Esposito asks, number one, how can you be sure that cysteine do not get oxidized? But I think you removed most of them, if not all. Do you think that an antiviral drug may be based on the strategy of dismantling the viral membrane? Okay, I don't follow the second question. Maybe you do, though.
I'm sorry, can you read again?
So the first was about cysteine oxidation.
Mm-hmm.
Do you have these cysteines left in the sequence, or did you change them to alanine?
No, we did not. We used the intact, including the old.
Okay.
And then we compared both reduced and non-reduced spectra, but we don't see any change, big change at all. So all the data for the full lengths are non-reduced, no reducing agent, but cysteines are there.
I see, thanks. I'll go on for the, if Gennaro is there, maybe you can raise a hand and later discuss.
Yeah, I think the second question means whether the drug can disrupt the membrane, and that would be the mechanism of action.
Meaning the lipid components of the membrane?
Mm-hmm. Yeah, exactly. I think there is a debate.
But I think I already showed the comparison of the full length and then the two truncated the data with the drug. So here, no significant change in the EC, but in the same condition, right? So that suggests that the drug mainly interacting with the protein, not the detergent or lipid. Yeah, here's the detergent, but yeah.
Then anonymous attendee asks if you've seen yet any differences between phosphocholine 16 compared to lipid bilayers.
That we are going to look into. We haven't we haven't looked at the data in bilayers yet, so that's definitely the next step. We will put into the bilayer and then compare the structure and orientation. Yes.
Okay, there's another short one. How did you confirm the molecular weight?
Molecular weight.
I guess it's in reference to the E protein.
Yes, the E protein runs as a monomer in STS page clearly, but in the native gel, their report with the other construct runs as a higher oligomer, the weight. So they suggest they form the oligomer, but we have to confirm that by using a various method to make sure its oligomeric state.
Thanks, and just a few more are left now. So one is, did you compare the peaks with the SARS-CoV-E protein for backbone assignments?
No, because it's a different, the environment. The previous results were obtained in DPC and DPC SDS mixture or LMPG. So all the spectra chemical shifts are slightly different. So it's difficult to directly compare. So we have to assign independently, completely in our experimental conditions.
One more, it seems that the binding of the side chain moiety depends on its hydrophobicity in relation to, I guess, the HMA. Would it be possible or useful to test whether the side chain moiety in a different context, not a channel blocker, binds to the same place?
That's a good idea. Yeah, that's also we can try. Yeah, it is definitely related to the hydrophobicity, not just the size. Yes.
There's then a question about phosphorus 31 calibration on slide 23. It's not around 0 ppm, question mark.
23 here.
This is an oriented sample.
Yeah, it is oriented. That's why the 0 ppm means isotropy, right? And the chemical shift around -10, -11 is the indication of the alignment of the phosphorus head group perpendicular to the applied its lipid bilayer perpendicular to the applied magnetic field. Yes.
Yeah, exactly. So you're reading out the alignment. One last question is still there. Did you add E protein and SP to see if disulfides form between the two proteins? And another addition to that just showed up. Do these proteins undergo post-translational modification?
That we haven't, we don't know yet. We haven't tried that yet.
For the disulfides. And for post-translational modifications, are the cysteines polymetylated or otherwise modified?
There is a literature, right? The E protein sequence cysteine is polymetylated, but because we expressed in E. coli, there's no post-translational modification here. But we actually, don't know about that yet.
Yeah, it's of course difficult to synthesize that sort of construct. Well, that's all of the questions that I see there, and there were no hands raised, so I'll turn it over to the next introducer. Well, once again, thanks a lot for the excellent presentation and the lively discussions.
Thank you.
So, hello everyone. We're very grateful that we can present our work on COVID-19 in Frankfurt and beyond. And we are a quite large team. So two of us will be presenting the science, and I'll just give you a brief flavor of the COVID-19-NMR consortium. So you have seen this picture that has been published in Nature 2020, and it actually summarizes the molecular components of SARS-CoV-2. As we know from sequencing, high similarity with SARS-CoV-1, more than 85%. And you see that approximately some 30 proteins are being made from something like 30,000 nucleotides. And when we saw that end of March, we thought we should start working on this, trying to distribute tasks and determine if missing all RNA and protein structures of the viral proteins, but foremost also generate chemical shift assignments that would support large-scale ligand screening campaigns by NMR. And we focused first on fragment screening, but then later on also repurposing drugs and alike. Our data were deliberately made publicly available weekly on the indicated web pages. Now, the consortium has grown. We have a core team of people in Frankfurt, but we have partners all over the world that actually, in a coordinated manner, work on the viral targets. That means we actually talk who's working on what, and we actually share proteins, we share RNA, we share the chemical shift assignments, and we, as said before, we have this COVID-19-NMR.de and .com with news, mission results, protein results, screening results. And once we obtain a new sample, for example, this RNA, we put them immediately on the web page such that somebody else who has the same sample knows that this is consistent with what we do and vice versa. And as a little show off, this is just in numbers what we did over the last 22 weeks. We have now 15 countries, some 170 people working, 4 papers submitted, in total 390 days of measurement time, 15,000 data sets, all in logs by the company Signals, 17 mRNA IDs submitted, and we produced something like 200 protein and RNA samples. And with this, I'm done, and I hand over first to Andreas, who talks about the protein part of the project.
Yes, hello everyone from my side. So let's get started with the proteins first. And as you had seen from Harald's introductory slide, this is where we are. The genome encodes for around 30 proteins that we can subclassify into 3 major groups, which is the 16 non-structural proteins that make up the replication transcription complex, followed by the what we call structural proteins, for instance, the ones that you just saw, the spike, the envelope, the membrane, and the nucleocapsid, which capsidizes the RNA. And a third group, which is the so-called accessory ORFs, relatively unknown proteins that have accessory roles in all the steps of viral replication. So the COVID-19 NMR consortium has decided to investigate as many of those proteins as possible by means of NMR spectra. And actually our initial strategy was based on the idea to exploit available structural information, which back then at the end of March was something like 150 PDB structures, mainly from homologs like the first generation coronavirus. And we then stepwise included remaining ORFs substructures, and we always used available homolog information that we could get. And the basic idea is to, first of all, define protocols for all the proteins that we purify. I will not talk about this in detail today. Followed by spectra and their at least partial backbone assignments before all that goes into an NMR-based screen, and eventually structures of proteins alone or in complex with the RNAs you are going to hear about afterwards. So what we also did is we also tried to cover the full functional context of the virus, which usually includes protein-protein interactions and protein-RNA interactions. So those 6 bullet points that I'm giving here in the middle are basically the central idea of what we are going to follow in terms of function. Saying that, this is the current status of the protein part of our project, and as Sarah has mentioned, this includes people worldwide, and I'm trying to summarize this here, and it also highlights the very basic oversimplified way of a pipeline going from protein design, construct design, through production NMR spectra to screening. And I would also like to mention that we currently do provide all the proteins that we make to external collaborators that work on the production of antibodies against all of the protein components of the virus. So this is basically the same shown in spectra. So I guess this is a nice overview showing, and it's actually not all of them, it's almost all of them, HSQCs of the proteins that we're looking at. And I guess you all agree that this is a very good starting point for follow-up work on and for individual screenings of proteins in-house and together with people all over the world. So all this is publicly available. And in fact, I show you one example from my own lab. This is the canonical route that we take from parallel design of constructs based on homolog structures through the optimization of expression and purification towards the assignment and finally the screenings. And I have to say that all our assignments are publicly available to be seen on our homepage and of course deposited in the BMRB, as Harald already said. And in fact, this is what we're trying to aim at for all of the proteins. And I would like to go into a bit more detail for one of them. So the protein you just saw, which is the NSP3E, is actually part of a much bigger complex. What I depict here in terms of domain arrangement is NSP3. It's the largest coronavirus protein, and it has two transmembrane helices. So it's a transmembrane protein, and in its cytoplasmic part, it comprises a whole bunch of nucleic acid binding domains. And what is shown in orange here is actually all the domains that back then came with structural information available. And the protein has recently been described in the context of a pore-forming complex. So it seems to be relevant for the so-called replication organelle, which separates the virus inside the cytoplasm. And the NSP3 is a perfect example of the basic idea and approach that the consortium takes. So all of those cytoplasmic domains are being worked on. For many of them, we do have assignments. And this is the perfect drug target in a way, because with all those domains and their unique functions within one big protein chain, this seems to represent a perfect drug target. And in fact, what we do know is that one of the proteins, which is the papain-like protease shown here on the right side, so it's one of the classical viral targets. It's one of the two proteases that the virus comprises. And this is an established drug target. And additionally, what we think is another potential drug target could be the NSP3B. The NSP3B is what we call a macro domain from a biochemical point of view. And what macro domains do is they do remove ADP ribose, monomers or polymers from proteins. So not surprisingly, we do see that our NSP3B does bind to ADP ribose with low micromolar affinity. And the poly-ADP ribosylation, in fact, is a very important post-translational modification and often occurs during immune response. So in case of D-ADP ribosylation, what we expect is to have a suppressive effect to host immune response. And we currently assume that not only the coronavirus, but also other viruses exploit this system to circumvent immune response in infected cells. So blocking the NSP3B macro domain might be an option to actually interfere with viral replication. What now gets interesting here in this context is that one of the drugs that has been recently approved is remdesivir. You might have heard of this. And remdesivir came into our mind because it apparently shows a similar biochemical or chemical building block based on nucleotide in its middle. So we use this drug to actually see whether this might also have an effect on the macro domain. And what we did first is we went into literature to see what remdesivir does, because what you need to know about remdesivir is that it undergoes intracellular processing after application, and it usually ends up in this triphosphate version, which is actually the one that inhibits the RNA-dependent RNA polymerase. So that's what it has been approved for. What we did is we used remdesivir and its triphosphate version and the so-called nucleoside version of the remdesivir, and we applied all of this to our NSP3B, and we found that the nucleoside indeed binds the NSP3B, showing nice chemical shift perturbations and an even higher affinity and even lower KD as obtained by ITC here. And indeed, that's not surprising, because if you compare the chemical structures at a appears very obvious that just by steric reasons, the smallest of those compounds might be best fitting into the potential binding pocket. So this is the affinities that we obtain from both NMR and ITC. We then succeeded in crystallizing the nucleoside of remdesivir, which I call NOR. And the crystal structure indeed shows that the protein binds the NOR compound within the same pocket as it would bind the ADP ribose. So this is a highly resolved crystal structure that shows the crystallographic dimer, while we know the protein, in fact, is a monomer from solution studies. And it comes with this very dense network of interactions with protein side chains, and it comes with an additional nitrile group that mediates interactions with those backbones, and it might be one of the reasons that the affinity is actually increased. And finally, to sum all this up, we also confirmed those findings in solutions. So what I'm showing you here is just one example of filtered NOSI spectra, where we, for instance, in this case, see the interactions or the close vicinity of the H2 group from the compound interacting with the backbone amides of those two amino acids. So altogether, what I have shown you here is that we have probably found a good starting point to also find an inhibitory drug towards the NSP3B macro domain. And with this, I would actually like to hand over to Boris, who's now going to introduce you to the RNA part.
Yeah, hello also from my side, and it's a pleasure for me to talk now about the RNA mission of our project, in which we have basically three aims. First, we set up to determine the secondary structure of the cis-acting elements of the viral RNA by NMR, then start to assign and try to solve high-resolution structure of as many as possible structurally or at least biologically interesting target RNAs. And last but not least, starting screening of these structured RNAs against fragment libraries to support the search for drug candidates. Probably most of you are interested more or less in proteins and not so much in RNA, but to give you an idea why at least I love RNA so much and why it's important. You know very well these pictures of the virus particle, and actually it's filled with RNA. The coronaviruses actually have the largest RNA genomes, and the size is roughly 30 kilobases. It contains a plus-stranded RNA, meaning that the RNA that is in the virus and gets released in the cell can be directly used as an mRNA, and the polyproteins are translated from this. And of course, it's also the template then for subgenomic RNAs, and also for basically the counter strand that is made and then again transcribed into a plus strand, which is then packed into new budding viral particles. And so this highlights that actually the RNA is really a central biomolecule of the virus and of high interest here. When we started the project, we first looked at the secondary structure prediction of the RNA, and decided to focus basically on three parts of the RNA, which is the 5' untranslated region, the 3' untranslated region, and in the middle of the translated region, a part that is responsible for frame shifting. And you see in comparison also to the SARS-CoV virus that there are kind of conserved structural elements, secondary structure elements depicted here that form these stem loop structures here. And they all are implied to have important roles in the transcription and in the translation of the proteins and the viral RNAs, and also especially the 3' UTR also plays a vital role in the replication of the protein of the virus. And so we wanted to study the secondary structure, and of course 30 kb is a bit too big for NMR, so we want to play our divide and conquer strategy and derived parts of the RNA that represent these structural elements and wanted to solve the secondary structures of these elements in the first place. Therefore, we set out to have these samples as 15N labeled samples. And then something kind of magical happened. We actually, within more or less 2 to 3 weeks, set up in making these different samples depicted here. And this, we are very thankful to have really put up a strong force in the RNA Biolab that is led by Anna Wacker and Julia Weigand, and they made it possible to clone all those constructs in plasmid, then do an in vitro transcription. Then we do standard and ion exchange chromatography. We purified all those constructs by preparative page. Then we included again an HPLC step to purify the RNAs from remainders of the page, folded it, buffered, dissolved it, and had then really nice NMR samples from this. And all of these samples then we recorded spectra, and for all samples, all these 15N labeled samples, we recorded standard H-noses, 15N HSQC and HNN COSIs. Some of the samples were also 13C labeled, not all of them, depending on the stocks we had in the lab at the time. And I just want to show an example. This is here the stem loop 4, where we were able to delineate really the secondary structure of the RNA and could assign all the peaks of the exchangeable protons, and for this case also we could assign the aromatics and the H1 prime protons. And I just want to show all these spectra because it highlights the beauty of the samples and also of the experiments that were run here. And the assignment of all of the RNAs was again a team effort where we put postdocs, PhD students into 2 or 3 people teams that took these spectra and assigned them. And so in total, we now have all these RNAs assigned, and a paper led by Anna and Julia is about to be published soon. We are in the revisions where we will publish all the assignments and all the secondary structure of these elements, and also compare this to DMS footprinting experiments. And as this is much too much to go into detail for each of the samples, but we will find all the spectra with all the assignment on our homepage, and also on the BMRB page, all the assignment is presented. So you can follow what we have done there, and you even can download the original data and the spectra of these RNAs I show you here. And but we didn't stop at the secondary structure of each of the elements, but we also looked at the larger assembly of these fragments, because of course there could be kind of tertiary interaction between those stable secondary structures. And as an example here is shown the first basically 180 nucleotides of the viral RNA, where we have solved the secondary structure of the individual hairpins, and you see if you overlay it, it basically perfectly fits on this part. And this is even possible for even larger constructs. So if we now look at basically 500 first nucleotides of the genomic RNA, this seems, first of all, it's for the size of RNA, it's a nice TROSY spectra. And if you compare that to the elements, you basically find all the peaks back here in this spectrum. With now having the secondary structure element of all these RNAs at hand, and also having in the meantime, unlabeled samples. We started screening efforts that is led here in Frankfurt by Sridhar Sriramulu. And we screen the RNAs against a fragment library containing 768 fragments, and we apply the screen to all of these. So this is a bit older of a slide now. Basically 16 out of the 17 different samples are finally screened and analyzed. And today I just want to go into detail what we have found for this pseudoknot here. This pseudoknot is a secondary structure that is needed for the frame shift, the ribosomal frame shifting. It's a -1 frame shifting, so it contains a slippery side and actually a stable secondary structure. And for coronaviruses, this is such a pseudoknotted structure here that is also found in the SARS-CoV virus. And so this structural element is really also very functional, important for the life of the virus. And so it might be it could be at least an important drug target. And now, as we have screening against not only this RNA, but also the other RNAs, we basically can discriminate what are unique binders towards this pseudoknot RNA structures. And in total, we find 11 unique binding fragments to this pseudoknot. Going from this, we collaborate with Marcel Blommers from a company called Saverna, who will do chem informatics and who will then suggest, or already did suggest, to improve the fragments and have larger fragment molecules which are more drug-like and which are also able to be modified in a way that we have now the specificity from the screening and can combine this then with known RNA binders to also increase the affinity here. And the molecules we have found for the pseudoknot as unique binders are shown here. And the same structural element was also addressed back in the days for the SARS-CoV, where they have identified this as a binder. And you can see the chemical similarity of our fragments we have identified towards this compound that binds to with high affinity or higher affinity to the pseudoknot from the SARS-CoV virus. So we are thinking that we are on the right track to find a molecule that really binds to the pseudoknot. And we of course now start thinking also testing this in vitro if it really stimulates or inhibits the frame shifting in the virus. And with this, I want to come to the end and thank all the people that are involved in this project. There are just too much to name them all. Special thanks goes to Sridhar, who's really the key figure also for the screening, both of the proteins and the RNA. Not here on the list, I have to say Anna Bakker, who drives really this RNA lab team and is basically responsible for having such a great pipeline of RNA production. And for the NSP3B story, this is a collaboration with Germ in Florence, Francesca Cantini and Lucia Banci, and also with the people from Patras. And for the crystal structure for the measurement time at the Bessie in Berlin. And not to forget also Julia, who is highly involved in making the RNA samples and also the biological testing, and also Marcel Blommers from Saverna, who do the chem informatics for our drug targeting studies. And with this is the end. And we are happy to take questions. And I hand over to Harald.
So Marcus, who is handling the questions? Am I handling the questions, or what's the idea?
If you would like to handle them, that would be great, Harald.
Okay, great. Do that. Thank you for the nice response. Voice is too low. We can't change it now anymore. When you show when you show the NH interactions between the drug and the protein, would then be a place for some water molecules. I hand this over to Andreas.
So that's a good question. In principle, the compound we're looking at here does not fill the entire pocket yet. So this is only a kind of starting point. And for the interactions I have shown, I would say no, not for those ones, but in principle, there are additional interactions possible through waters. I would say yes.
Then comes question from Fred Lamberger. Would it be feasible but advantageous to identify drugs that affect the function of multiple RNA targets of the virus? Hand this over to Boris.
So, I mean, we see small fragments that at least bind to different RNAs, but of course, fragments are not drug targets. And if we now want to increase also the affinity, we might lose, or we probably will lose, that it binds to very different RNAs. And I don't know if it's a good drug that binds, if that would be considered as a good drug that binds to more than one RNA specifically, because then also could also bind then to similar structures in RNAs in the host cell.
Next question, I dare to answer myself. Would you expect more specificity in binding RNA from the ternary interactions rather than secondary interactions? First, binders actually need non-canonical parts of the RNA. In the canonical parts, you may intercalate, but there's simply no chance to achieve specificity. Along the lines, yes, ternary interactions could be targetable. The way we try to address this is that we screen the fragments, the individual isolated segments, and the longer constructs, and try to see overlap or what appears only in the longer constructs. One of the issues that some of the guys that have done screening already is that with this so-and-so affinity that you have, just to get the binding site from NMR is not at all trivial, because at these affinities we are at right now, it actually does take a time to map the binding sites. So this is not so clear. For the protein example, there we did see slow exchange at something like 2 or 3 micromolar affinity of drug to target. But with hundreds or 200 micromolar, it's not yet really that we can really nicely titrate the binding site, even if we have the assignments.
Harald, do you allow me to ask a question?
Please.
Christian. So I was wondering, do you have any sense of what affinity you have to reach in order to make an impact on treating people?
I don't know the answer to the question yet. I mean, the first, on the NSP3B, we have a collaboration where people have viral models. And so we have submitted the first molecules and see whether they do something. This remdesivir, if it only binds or if it only inhibits replication, it's an irreversible inhibitor. So there the affinity is not so clear, because it's, you know, it's replication inhibition and irreversible inclusion of the into the synthesized chain. So I mean, I would say typically, as always, in the low nanomolar regime, right?
I mean, I looked for the pseudoknot. I looked in this Jack's paper, and there, I think they had an affinity on IC50 of 0.8 micromolar. And I was wondering, where are you now with the inhibitors that you found? And the other thing is, I mean, is it feasible to put them maybe together in a phasic-like approach that they attack different?
Yeah, Christian, second question first. This is what we do. The fragment library had been developed by friends in Diamond, Oxford, as a poised library that is geared towards easy follow-up chemistries. If you look at these fragments from a chemical point of view, you can easily link things. Yeah, and the idea now is, although this is not yet proven, but this is what we follow, is that we have the specificity of by the fragment, the binding specificity by the fragment, and we add affinity tags, so to say, to increase affinity. And affinity tags can be derived from aminoglycosides or from aromatics. I mean, these are the two bets that we have right now. And we do synthesize this compound that was initially identified in Jack's, this number 43, not 42, 43, is not anymore available. So Michael Goebel is synthesizing that for us. And so we basically now try to get in contact also with medicinal chemists to say, look, these are our first proposals. Are you not interested in following up? Because we cannot possibly do this all by ourselves, because the chemistry is, is a big thing. Somebody anonymous asked, how much funding did we receive for the whole consortium? For the whole consortium, unfortunately, I don't know that there is a funding a system that would fund the entire consortium internationally. For us in Frankfurt, we were extremely fortunate that the DFG actually evaluated our first grant within like 2 weeks, 2 or 3 weeks. And we got a half-year funding that was in particular valid for the isotope budget, because we had the funding to actually send the isotope labeled proteins then worldwide. And we also purchased the library, which we can also send, or it can actually be purchased from a vendor. We're now, and maybe somebody is in the audience who had a chance to review the grant applications, and we're very grateful for the very supportive evaluation. We're currently reapplying for German money, but all of this is German money, and we were also happy to receive funding for automation, so spectrometer automation. So we have 2 spectrometers that run day and night for the screening now. So we have dedicated 2, and before we had 1, and now we have 2. And again, this was evaluated within 4 weeks, again by DFG. But I have no idea, and I'm very, very sad that there is no idea how to get international funding. We tried to apply for the Gates Foundation. It took them like 40 minutes to turn our application down. So we are not clinical enough. So most of the money from the national consortia go into direct clinical application stuff and not fundamental research. Are there any PROTAC systems being developed along with the NOR drug? Good question by Grace. I don't know. I don't know. A general question on remdesivir, Anna De Angelis. Is it known how much reaches the endoplasmatic reticulum or the whatever, ergol? Is there a known trafficking mechanism? Thanks. Yes, there is a paper in Feb or March of this year that actually talks about the real PROTAC mechanism. So everybody thought that the PROTAC would be the part that actually traverses the outer membrane. This is not the case. This is only the case if you do cell assays. If you actually go into blood, then in blood, remdesivir is already hydrolyzed to the nucleoside, and it's the uncharged, not phosphorylated nucleoside that is being incorporated in blood, in serum, into into human cells. What does it mean consortium partner arrived from and consortium partner shipped to Gennaro Esposito? So we sent proteins, for example, to Florence. We expressed 13C15N labeled protein, sent it to Florence. We expressed protein for other partners. And this is the day when we sent it by FedEx or others. And the courier actually services are currently also slow. So it's also not even easy to ship stuff. And we sent RNA to Weizmann and to Stockholm and to Valencia. So this is meant with shipped and received. So it sometimes takes 3 weeks to actually get a sample from A to B nowadays.
Also, I have a curiosity. So the RBD spectrum that you showed, which is a very important protein in this system, was it the glycosylated protein? Because it turns out that this protein actually expresses better in the glycosylated form. I think it's very insoluble, the
Oscar, you're absolutely right. We did not yet did not yet go into eukaryotic expression. I mean, some of the stuff expresses like crazy, very nicely, and the ORFs, for example, express very badly. So I mean, we did do cherry picking, but you're right. Some of the stuff will eventually have to go into more complicated eukaryotic expression systems.
Another question showed up in the answered questions as a response. It says, does the SARS-CoV-2 have a VPG-like protein that links the RNA? The poliovirus and some other viruses have this currently.
So
does it have a VPG-like protein linking RNA?
Not that we know in detail, no.
We follow it up. We have homework to do. We don't know.
It's a Sid who asks.
Okay. Sovosbuvir is also a target drug molecule. Consider it. Thank you. We will do. Just to follow this up, there are something like 43 drugs that target viral proteins. And there were repurposing campaigns already running. So this is yet another strategy that people apply.
Harald, I would also like to ask something. So for the NSP3 protein, you look at all these individual domains. I mean, how much higher order structure is there between the multiple domains, and could that be relevant? Or?
Yeah, so actually for the NSP3, this is a very good question. We have absolutely no idea in what way, whether and how all those domains interact with each other. There's a few number of structures that shows at least 2 or 3 domains together. And those are basically usually classified into one subdomain, for instance, the NSP3C. But in principle, we have no clue. So the NSP3 is a very, very specific thing. So it seems to align all those domains on a chain, and it might, for instance, hand over the capsidized RNA to the pore. This is just one possible hypothesis. So it's a lot of low affine RNA interactions. What and how they interact with each other, we don't know. There's in fact not much known about the interactions between NSP3 subdomains, but rather with other NSPs in the virus.
Okay, thank you.
There's one more general question. How many of non-structural proteins are methyltransferases? One, no?
There's only one methyltransferase, but it interacts with other subdomains, so it doesn't act on its own.
So no more questions. I give back to Markus.
Well, maybe I could ask something for those of us who aren't so close to drug screening, if you could provide a little detail on the workflow. I mean, for example, how do you decide when a hit is there? Do you first do CSP? How many do you do affinity for? And do you ever do a divide and conquer type approach where you test multiple compounds at the once? These kind of things.
So we, so I give some of the difficulties And first of all, we have a common RNA buffer system and have assigned all the 768 compounds to within this buffer system, which is one of the steps. For the proteins, it's not always easy to always have the same buffer system, because proteins actually have better stability in different buffer systems, which then in turn means that some of the ligands you have to reassign, which is actually tedious. And then the 768 ligands, I think, are subdivided in 64 mixtures of 12 compounds each. And we conduct proton screening and also fluorine screening. In the fluorine screening, out of the 768 compounds, 102 or so are fluorinated. The fluorinated compounds in particular have the advantage that you can very easily get KDs because you can titrate the fluorine signal. For others, I mean, fluorine is, up and down, and many people talk about the advantages of fluorine. This is so and so. The protons is also great. We do STD, we do T2 spin locks, and we look at chemical shift perturbations. And basically, one of the things is, as the target size actually differs from target to target, you get different T2 effects on basically, if the target is large, your T2 is more affected than if the target is smaller. So you have to recalibrate this. Then it's actually first we use Bruker software and MNOVA software to analyze this, but it's also a specific team that analyzes all the screens. And in principle, you have to rescreen then the hits, because of course it is a difference whether you have 12 ligands in a mixture, because you also have various unspecific effects at these affinities. So, and the RNA or the protein is 10 micromolar concentrated, and we have 20-fold excess of ligand over protein or RNA. So, and it takes, one screen takes 3 days. I mean, screening one library takes 3 days.
Thanks.
So may I follow up this? So, I mean, at these ratios and as you said, affinities, I mean, do you not see for every 12 compound mixtures some effects?
It's quite striking that the number of hits are very different. I mean, we had NSP5 and we have a ton load of hits for the main protease, but we also have other proteins that have barely any hits. So this is also said by Marcel Plommers, who used to work with Novartis, as many of you know, that the fragments do help in assessing the general trackability of a target. So you get an idea whether a target is in general trackable or not. And with NSP5, we have then taken the hits, and you then take the hits and you screen those hits cheminformatically against all available chemical space and some chemical structure bases, chemical databases or so. And then you get 5,000 alikes. Yeah, I mean, you have 50 hits, you get 5,000 compounds that chemically look similar. And then you prioritize how many of these alike hits do you buy. And then you rescreen based on the initial fragment hits. And then with Tassos Periakis at Amsterdam, we do thermal shift assay and follow up there to have a secondary other biophysical method to screen the NMR hits. And for NSP5, sorry, I go and talk, but you interrupt me. For NSP5, the diamond also has screened the same library by crystallography. And we find some of the hits also that diamond finds. We find more hits. And we are very happy to say that all the hits we find by NMR also have a thermal shift assay, which is not for all the hits the case. So this re-validation of the NMR hits is necessary. And at that point, we could start now chemistry, but then comes the question, NSP5 is competitive like crazy. It is the main protease, and it's a cysteine protease. And people in industry say cysteine protease, targeting cysteine proteases is a nightmare. is not an easy drug target. Now, you ask many people, you will get different answers. So, but these kind of things have to be then looked at, and it enters medicinal chemistry, and we all know that this is a long run, and you know, and this is of course not where NMR is best at. But I mean, we try, but we can not do this for all the targets. So we are happy that others would say we would take your initial fragment hits and would be happy to follow up these initial hypotheses, because we cannot possibly do all of this.
So, and I mean, because you mentioned the thermal shift assay, I mean, could you just do the thermal shift assay, not do NMR? I mean,
me, Markus, me not.
Okay.
Do you also do competition assays once you have a hit in order to put a threshold on the KD that you are looking at?
Yeah, we have, there are examples where we do this if you have a cognate ligand, but we wouldn't be yet there. But for the pseudo-knot, we would probably be in a position that there's a sort of good idea, initial ligand, and then we can
or one of the hits that you found. Like
exactly.
Mm-hmm.
I have another question. How long does the pandemic has to last that something that is developed at this stage will be useful to humans?
Christian, I think this is the wrong question.
My answer
to this question is the following, Christian. If the scientific communities had done more research on COV1, which is more than 85% similar to COV2, we would be in a way better position to combat COV2. And so what we do is actually good for COV3.
Okay, that was actually my my question, whether the next one could again be a coronavirus, like the first one, the MERS, and now this one, that one should really go through with it. Of course, that would mean that it would be great to compare, for example, targets, similarity. For example, I mean, one could imagine that if that the probability to experience for COV3 the same target as for COV2, one could extrapolate from comparison of COV1 with COV2.
Yeah,
and single out those which have maybe larger changes or something like that. Of course, it's just it's a guess, but
for RNA, for not translated parts of the RNA, this is the most stringent question, to compare the similarity or identity of untranslated regions of RNA between different viruses, because you don't even have to express the stuff, right? And even there, these beta coronaviridæ have enormous identity. Mm-hmm. So they don't change. So I don't know whether it is understood how we can reach immunity to something, and then the other one is barely different, and we are not yet immune against this. But for this part, we discussed this a lot when we set up our consortium. We were convinced that given this high identity of these coronaviridae, it is a valid argument that that we could do something that will help later on. And if you see how long it takes to vaccinate this, and we don't know whether they work or not, or how effective they are, if we had a starting drug that we could give against coronaviridae, that would be actually fantastic. And by the way, remdesivir, as you know, it can only be applied in very, very severe cases of the thing. In very late stages, it is allowed to use this, because it's of course very unselective.
Which I mean, I don't see as a good reason why it could not be. I mean, I never understood why it is not given early when the replication is not so strong. Because I mean, these days one has a lot of predictors about the clinical progression, and much more than one had in March. And so these days, I think one can more easily predict who will have a severe progression and who doesn't. And so that is probably just a delay in the from the regulatory offices or something. But yeah, whatever. I mean, I'm also not an expert in that.
There's also a comment in the Q&A, right, which a little bit relates to this, right? I mean, so probably one has to do some political activity. So when the SARS-CoV-2 pandemic is somehow gone, to convince people to continue this research or not wait until the SARS-CoV-3 starts.
There are further remarks in the questions and answers. Harald, do you want to read them?
Yeah. John Bon Xiong, that is correct, but the need of SARS-CoV-1 disappeared when the first SARS epidemic died down. We had discussed this. Then Emmanuel Verroutis makes a very nice comment. The unfortunate circumstances led to an exemplary collaborative effort that otherwise would have required several years to achieve. This is correct, and I'm very grateful to the support, worldwide support that we have seen. And I mean, one of the things why we are also grateful to talk here is that we also want to invite each and every one of you who would like to participate, and they sort of get in contact thing on the web page. So send us an email. We have followed every email that has been sent to us.
So maybe then we close the official part. Thanks again to both speakers for absolutely fantastic presentations. Great work. And so next week we have again two great speakers, Anja Böckmann and Pramod Vallupari. So please tune in next week. And if you want to hang on, we're still here for a few more minutes. Thank you.